Hello everyone. Today we will discuss about a short note in biochemistry, that is amino acids. Amino acids are structural unit of proteins. The proteins are linear polymers of L-alpha amino acids which are linked by covalent peptide bond. We will see what are the alpha amino acids, what are L-alpha amino acids, but first of all we'll see the general structure of amino acids. So here is the general structure of amino acids. It contains R group, amino group. In addition to this, it has hydrogen group and one R group, that is side chains which are attached to this carbon atom. In this, both amino group and carboxylic group attached to the same carbon atom, and so these amino acids are also called as alpha amino acids. This R group, that is side chains, differ in structure, size and charge. So this is the general structure you can see over here. It is also called as the ionic state of the amino acids. So this is the ionic state in which this is the alpha carbon atom. Alpha carbon atom to this carboxylic group is attached, amino group is attached. So these are the chiral centers. Alpha carbon atom. This alpha carbon atom attached to 4 different groups, and so it has the chiral center, and because of this tetrahedral arrangement of alpha carbon atoms, the 4 different groups can occupy 2 unique spatial arrangements, and thus amino acids have 2 stereoisomers which are optically active and mirror images of each others, and so they are called as enantiomers. So these are called the enantiomers. Now amino acids are of 2 types, in which that is levorotatory, that is L amino acids, and dextrorotatory, that is D amino acids. L amino acids, where L stands for levorotatory, rotating the plane polarized light to the left, and dextrorotatory, rotating the plane polarized light to the right. So these are based on absolute configuration of 3 carbon sugar glyceraldehyde. So this is the structure of L-glyceraldehyde, and this is the structure of D-glyceraldehyde. Now amino acids in L-glyceraldehyde, hydroxyl group is present on the left side. Here you can see hydroxyl group is present towards the left, and in dextrorotatory, hydroxyl group is present towards the right side. In the same way, amino acids, amino group is present towards the left side, and in D amino acids, amino group is present on the right side. L amino acids occur in all biological protein, and that is why proteins are the polymers of L amino acids. Now amino acids are of 2 types, standard amino acids and non-standard amino acids. So standard amino acids, there are 20 amino acids which take part in protein synthesis, so they are called standard amino acids. And non-standard amino acids, the amino acids which these are not involved in protein synthesis, but they play a role in different amino acid derivatives and in non-protein amino acids. Based on this, the amino acids are classified in these 4 types based on structural classification. Second one is based on polarity. Third, based on metabolic fate. Based on nutritional classification. So we'll study the classification step by step. First of all, we'll study the structural classification. There are 7 types, in which first one is aliphatic R group, side chain with hydroxyl group, side chain with sulfur group, side chain with acidic group, side chain with basic group, and aromatic amino acids, and last one is amino acids. So these are the 7 types. Here we'll study now first one by one. So aliphatic R groups. So in this group it contains glycine, alanine, leucine, valine, and isoleucine. Glycine, alanine, leucine, valine, isoleucine. So this is the structure of glycine. Here is the alanine, here is the valine, leucine, and isoleucine. So these are the structure of all these amino acids. It contains, now glycine, here this structure of glycine, here it does not contains alpha carbon atom because it is not attached to 4 different groups. It contains only the hydrogen atom. It is only the amino acids which is optically inactive and has no chiral centers. Other all are active amino acids, but only the glycine is inactive, optically inactive, because it has no chiral centers. It has only 2 carbon hydrogen attached, and so among all 20 amino acids, this is only optically inactive. This is the structure of alanine, valine, leucine, and isoleucine. Second type will see side chain with hydroxyl group. This is serine and threonine, in which hydroxyl group, here you can see the hydroxyl group of in serine and threonine. Serine, threonine, and tyrosine, in this hydroxyl group, these are able to give their proton because of their alcoholic nature. Side chain with sulfur atom, here you can see it contains cysteine and methionine. These are the structures of cysteine and methionine, you can see over here. Cysteine, it contains SH group. See here, you can see the thiol group, you can see SH group, and which is acidic in nature, and so cysteine form a disulfide bridge. So in this way it forms a disulfide bridge, you can see over here, hydrogen atom. So these are the two disulfide, two sulfur atoms which are attached over here, and so it forms a disulfide bridge. Next we'll see side chain with acidic group. So it contains aspartic acid. We can see this is the structure of aspartic acid. It has, and its amide group is aspargine. Here is glutamate and glutamine. These are the structures of glutamate, glutamine, aspargine, and aspartic acid. These carry more than one carboxylic group. Now next we'll see side chain with basic group. It contains lysine and histidine, in which it contains one more amino group, and in histidine contains an imidazole group. Here it contains one more amino group, and here it contains an imidazole group. Next we'll see the aromatic amino acids, in which tyrosine. Here first of all you'll see the structure of phenylalanine. It has an benzyl phenol ring. This is the structure of phenylalanine. Here tyrosine and tryptophan, they participate in hydrophobic bonding, in which hydroxyl group of tyrosine and nitrogen group of tryptophan, hydroxyl group of tyrosine. So this is the structure of tyrosine. The hydroxyl group of tyrosine and amino group of tryptophan or nitrogen group of tryptophan, they are slightly polar, and it contains an indole ring. So this is the indole ring of tryptophan. This is the benzyl ring of tyrosine, in which hydroxyl group is over here. Tyrosine, tryptophan, phenylalanine, they participate in hydrophobic bonding, and hydroxyl group of tyrosine and nitrogen in tryptophan, they are slightly polar in nature. Next we'll see the amino acid group. Proline does not contain amino group, it contains the amino group. So it do not contain, and amino group NH3 or NH2, it contains a amino group. So this was the structural classification. Now as we have more 3 types based on polarity now. First we will say structural classification, then now second one based on polarity. So based on polarity, they can be in hydrophilic in nature or in hydrophobic, that is hydrophilic, that is polar, hydrophobic, that is non-polar. Based on structure, now here in hydrophilic, here you can see these are negatively charged, positively charged, and uncharged amino acids. So in negatively charged, which are the amino acids? Aspartic acid and glutamine, these are negatively charged. In positively charged hydrophilic, or we can say the polar amino acids, these contain lysine, arginine, and histidine. And in uncharged amino acids, which are the uncharged one? Serine, threonine, tyrosine, aspargine, glutamine, cysteine, and glycine. So these are hydrophilic, that is hydrophilic in nature, that is polar or water soluble, we can say. Now in hydrophobic, these are non-polar in nature. These contain these are also aliphatic in nature. So alanine, valine, leucine, isoleucine. And aromatic in nature, which one are the aromatic amino acids? Phenylalanine and tryptophan. So these are the hydrophobic, which are water insoluble amino acids. So based on polarity, these are polar and as well as non-polar in nature. So again we'll see the review of this hydrophilic amino acids: negatively charged, positively charged, and uncharged. In that, negatively charged we contains only two amino acids, aspartic and glutamine. Positively charged, lysine, arginine, histidine. Uncharged, serine, threonine, tyrosine, asparagine, glutamine, cysteine, and glycine. And overall hydrophobic, these are water insoluble in nature. So we will see alanine, valine, leucine, and isoleucine, which are aliphatic in nature, as these are non-polar one. And in aromatic, these are also non-polar amino acids, phenylalanine and tryptophan. Next we will see based on metabolic fate. So this group contains amino acids which take part in synthesis of glucose and fat. So these are called as glycogenic, we can say. So those amino acids which take part in synthesis of glucose, they are called as glucogenic. So these are alanine, asparagine, arginine, cysteine, glycine, glutamine, histidine, glutamate, methionine, proline, serine, threonine, valine. So these all amino acids which take part in the synthesis of glucose. So these all are called as glucogenic amino acids. Now ketogenic, those the amino acids which take part in synthesis of fat, so they are called as ketogenic. So this was the synthesis of glucose, in ketogenic synthesis of fat. So these amino acids take part in synthesis of fat, so they are called as ketogenic. Only two take part, that is leucine and lysine. Now apart from this, the amino acids which take part in synthesis of both, that is glucogenic also and ketogenic, that is glucose and fat. So these amino acids are called so these amino acids are called glucogenic or ketogenic. So there are four amino acids: phenylalanine, tyrosine, isoleucine, and tryptophan. So you can shortly call as PIT, P-I-T-T, that is phenylalanine, tyrosine, isoleucine, and tryptophan. So based on metabolic fate, these are glucogenic, and ketogenic, and third part which take part in both synthesis of glucose and fat. So these are glucogenic, ketogenic. Only these four take part: phenylalanine, tyrosine, isoleucine, and tryptophan, P-I-T-T. You can call this as a short form. And last, based on nutritional classification, these are essential amino acids, and in second one, non-essential amino acids. First of all, essential amino acids, the amino acids which are not synthesized in the body, they are called as essential amino acids. They should be taken through the diet. So they are valine, isoleucine, leucine, tyrosine, arginine, leucine, threonine, histidine, lysine, methionine, phenylalanine. And vital dilimb, it's a short form to just to remember all these amino acids. Vital dilimb, that is valine, isoleucine, tyrosine, arginine, leucine, threonine, histidine, then lysine, methionine, and phenylalanine. So vital the lymph. So these are the amino acids which are not synthesized in our body. We have to take through only diet. And the non-essential amino acids which are synthesized in the body, and they are called as non-essential amino acids. These are alanine, asparagine, cysteine, glycine, glutamate, proline, serine, threonine, tyrosine. So these are the non-essential amino acids. So all these amino acids, based on polarity, based on metabolic fate, structural classification, based on nutritional classification, all this contains only 20 amino acids, and these are classified on these all four bases. So this was the short video on amino acids, their general structure and their classification. So based on this, we have studied amino acids which are classified in 5 types. Classification of amino acids in nutritional classification based on metabolic fate, their structural information. So this was a short video on this. In next